Yes, good afternoon again. I'm Fernando. And first I want to thank the co-author whose collaboration was critical for the completion of this project. And in this time, we're going to talk about the nutritional effects of black soldier fly larvae protein inclusion in the diets of juvenile red drum. And I'm going to start my introduction with a now, telling you what it's actually happening now. So we can see that seafood consumption is increasing over the years, and we have estimated that that seafood consumption will go up to 23 million more tons by 2030, and that an aquaculture industry will be the one that will suffice for such a high demand. However, is this aquaculture industry the one who feeds itself from a fish meal? And it is understandable. Fish meal holds a high crude protein, excellent amino acid profile. It's much palatable and digestible. However, such a fish meal reliance, it's globally accepted as an unstable practice, promoting at the end fish meal prices to go up and market volatility, which at the end of the road will negatively influence fish farming. Therefore, alternatives to replace fish meal are paramount. So alternatives such as plants, animal products, insects offer both opportunities and challenges that challenges as general as the nutrient content or digestibility, or challenges as particular as the protein source. In this line, black soldier fly larvae is an excellent candidate, primarily because its capacity to convert food waste into high quality protein with a high content in the protein and minerals, and also presents a well-balanced amino acid profile. In addition, it's an ingredient that is easy to incorporate into the aquafeed formulations. Red drum is a commercial and recreational important species. It holds a high growth rate and high environmental tolerance, which makes it also a good candidate for fish farming inland and cage of offshore caging culturing. So evaluating alternatives to replace fish meal those alternatives can be also sustainably produced is critical for the fish farming of this species. So to this end, the objective of this project was to determine the effects of replacing fish meal with black soldier fly at different inclusion levels in the diets of juvenile red drum. And we will focus on the final ways, on the fish meal inclusion, on the growth performance, and also on the proximate composition of the diets and of the fish itself. Here, the experimental design, we formulated 5 diets. The first diet worked as a control with 100% fish meal with 0% black soldier fly meal inclusion. The rest of the diets contain an increasing amount of black soldier fly replacing fish meal with a 25 inclusion, 50, 75, and a diet with 100% black soldier fly meal inclusion totally replacing fish meal. The experiment consisted of an 8-week duration, and there were 4 replicates per treatment. Here are some of the HPO team during the daily routines, during the feed production, during the sampling, and during the lab analysis. So we get to the results. This graph shows the effects of replacing fish meal with black soldier on the final weight during an 8-week trials. Different columns with different colors represented different treatments. And if we focus on the orange columns, we can see that from week 2 until the end of the experiment, those orange columns represent fish fed 100% black soldier fly meal, and those fish fed that diet started to lag behind on their weight. If we focus on the week 6 and on the week 8, we can see that orange columns and the green columns, meaning the 25% inclusion and the 50% black soldier inclusion, presented statistically the same weight as the fish-fed control diet in red. So inclusion, this graph suggests that 25% and 50% black soldier fly inclusion is an appropriate replacement for fish meal. Well, a second degree polynomial function best fitted our data to predict the relationship between black soldier fly meal inclusion and fish weight. And that relation is a negative relationship. And by this, we mean that when black soldier fly meal is included, when we include black soldier fly meal in the diets, and when that inclusion increases over an acceptable limit, the values on weight gain diminish. And what is that inclusion limit? And what is that acceptable inflection point where the polynomial function predicts maximum black soldier fly meal inclusion for a maximal weight gain? The polynomial functions tell us that that maximum inclusion is 28 black soldier fly meal inclusion to replace fish meal if we want to yield a maximum weight gain of 2.3 kilograms per mass body weight. Polynomial function wise, that's it. That's the maximum inclusion if we want to get a maximum weight. However, when we talk about fish production, we normally talk about a range, an appropriate range of inclusion. And by that, we mean a range in which including black soldier fly meal to replace fish meal will have no detrimental influence on the fish weight when we compare it to the control. In this way, we can see that fish fed a 55% black soldier fly meal inclusion will yield the same weight as fish fed a controlled diet with 0% black soldier fly meal inclusion. If we take a look to the growth parameters, we can see that also fish fed 25 black soldier fly meal inclusion presented highest specific growth rate, best food conversion ratio, and highest protein efficiency ratio, which correlates with the data from the polynomial function prediction at 28 black soldier fly meal inclusion. If we dive into the amino acid content of each of the diets, we can see that for higher inclusion of black soldier, the amino acid content of the diets vary and most of them decrease. Such an amino acid content decrease suggests a correlation or relationship with the weight gain variations. So to determine if there is a relationship between that amino acid content and the weight variation, if there is any, and to which extent that amino acid content is responsible for that variation, we did a polynomial regression, a quadratic function predicted such a relationship. And the R-squared predicted the degree of the responsibility to which this amino acids variation is related to the final weight changes. We also calculated the estimated requirement for red drum. And we can see that when we increase the inclusion of black soldier fly meal into the diets, that amino acid content grows farther apart from the actual estimated requirements for red drum, therefore suggesting that diets have a deficiency in amino acid content. Finally, if we took a look into the fish itself to respect to the different treatments, and we saw that there was, when we took a look to the approximate composition and the amino acid profile of all treatments, we saw that there was no significant changes, except for taurine, whose concentration diminished as the black soldier fly inclusion increased. probably this is a subject for future experiments on taurine supplementation. And in conclusion, this study shows that black soldier fly meal can effectively replace fish meal in red drum diets, and that black soldier fly inclusion replacing fish meal ranges from 1 to 55% in the diets of red drum, and that black soldier fly inclusion does not change final whole body fish composition of the amino acid profile. Lastly, this is some of the work that we are still there is some progress, and we have some results on histology, on the effects of black soldier fly on the histology, and also some results on the palatability that you can see them as a poster outside. And I also want to thank everyone involved in this project, especially Duke Energy, Stratium, and the USDA RAS program, and all the people who participated during the maintenance and the sampling. Thank you.
So we have a couple minutes, so.
Yes?
What was the inclusion level of fish meal on the controlled diet?
No, in the controlled diet, it has no fish meal inclusion. It's basically a diet with fish meal only. Anyone other question?
Right there and then over here.
Great presentation, Fernando. Was the black soldier fly meal that you used, was it defatted or was it the regular?
It was, I think that Radu is in my take, I think it was defatted. Yeah.
49%.
Yes, exactly.
And when you were replacing the protein for the fish meal and were you doing that in a nitrogen basis or?
Yes, yes, when I was formulating the diet, so that I was formulating on the nitrogen basis, so they all can have the same protein content. Yeah.
The control diet was a fish meal-based diet.
Yes. Normally, a diet when you formulate is not only fish meal; it contains a couple of at least three sources of protein. So it also has soy protein too, has poultry too, but the main one is fish meal. Yes.
I may have missed this. What size were your fish when you began the study? And also, how come only 8 weeks versus a longer duration?
Yeah, we choose, I don't remember exactly the time, it's probably in the pictures. And we choose 8 weeks because at 8 weeks already in protein is a good time for have a reaction. For the previous experiment for carbohydrates, for that we took 10 weeks because carbohydrates show more time to show up. So that was the reason.
All right, well, thank you.
Thank you.